This summer, I'm going to be researching the GC1QR receptor and its role in cancer and tumor metabolism. Cancer, also known as malignant neoplasm, is a group of various diseases that all involve uncontrollable cell growth. According to the National Cancer Institute, the estimated cancer prevalence in the United States is about 12.5 million in males and females, about 5.8 million males, and 6.7 million females. This figure here shows the total number of new cases of various types of cancer in both men and women. In 2007, there were less than 750,000 cases of cancer in men and almost 700,000 cases in women. The GC1QR receptor, shown as the green ovals on the surface of the cell, is also known as the GC1Q binding protein, P32 or P33. It is a multifunctional cellular protein that interacts with components of the classical pathway of the innate immune system or the complement system. It is located predominantly on the mitochondrial matrix and other cellular compartments, including the endoplasmic reticulum and nucleus, as well as the cell surface. The receptor has been linked to many bodily functions, three main ones being inflammation, infection, and cancer. The receptor was first discovered as a receptor for C1q, an important molecule involved in the immune process of clearing pathogens out of an organism, as seen in the figure. More research has been done with the receptor and its role in inflammation and the immune system, but I will most probably be doing research on its role in cancer, which has recently been highlighted. In a study done by Fogel in 2008, researchers looked at markers for cancer which usually target particular cellular components that are unique to tumors. In this study, scientists looked at LYP1, a tumor-homing peptide that has been shown to provide a marker for vessels on tumors and also bind to these cells and inhibit tumor growth. In this study, researchers found that the GC1QR receptor was a receptor for LYP1 and a potential marker for the peptide. As seen in the top panel, a blocking of P32 or GC1QR on the right induced less LYP1 binding to tumor cells as shown through phage staining. Also, as seen in the bottom panels, GC1QR signaling was much higher in tumor cells as compared to the spleen and kidney on the right, where P32 expression is often very high. This suggests that expression of P32 is a particular characteristic of malignant cells. The researchers in the same study also looked at human carcinoma cells. As seen in the figures, there was a much higher GC1QR expression as seen through red staining and many different kinds of tumors. These findings make cell surface P32 an attractive target molecule for the possible delivery of diagnostic probes and therapeutics into tumors. In a study done by Gabrahowit in 2004, researchers also looked at expression of GC1QR, but this time with more specificity. Scientists focused on breast carcinoma cells. This figure here shows the graphical results of a northern blot done to look at expression of GC1QR in breast cancer tissue. The first three bars represent normal control tissue, while the fourth bar represents hybridization in a breast carcinoma cell. The levels of P32 expression are much higher in breast cancer tumors. This elevated expression of GC1QR can also be seen in the two images. In Figure A, a breast cancer tumor is shown with immunostaining done to highlight P32 expression. In Figure B shows a regular breast tissue sample. While both samples do express P32, the staining shows a much higher expression in the tumor. While noting elevated levels of GC1QR expression in tumor cells, researchers in the study looked at knockdowns of P32 in breast cancer cells. The study found that unlike previous notions, a knockdown of P32 creates a shift in cancer cells from aerobic to anaerobic expressions. In the top two figures, it was found that the P32 knockdowns represented by the gray bars induced higher levels of lactate production and glucose consumption, both key activities that take place during glycolysis. Additionally, as seen in the lower figure, a knockdown of the protein induces a decrease in oxygen consumption, an activity that takes place during oxidative phosphorylation. This suggests that P32 may be required for efficient ATP production through oxidative phosphorylation, and the loss of P32 shifts energy metabolism towards glycolysis. It is also interesting to note that the elevated glycolytic rate and lactate production did not in turn lead to increased cell growth of the knockdown cell. As seen in the figure, as compared to a control cancer cell, a cell with a knockdown of the protein exhibited a lower tumor growth rate. This deletion of P32 expression increased glycolysis while reducing cell proliferation. Much of the research that has been done so far on the GC1QR receptor has shown that the receptor is not only an important regulator of tumor metabolism, but is also a promising molecular target in tumor diagnosis and therapy. The northern blot is a technique used in molecular biology research to study gene expression by detection of RNA in a sample. Northern blotting allows for one to observe a particular gene's expression pattern between tissues, especially during treatment. The protocol starts with an extraction of the total RNA from a homogenized tissue sample or from cells. A nylon membrane with a positive charge is often used for these procedures, and the RNA samples are most commonly separated on agarose gels. Hybridization probes are then used complementary to part or to the entire target sequence. Scientists are then able to visualize RNA on X-ray films. Flow cytometry is a laser-based biophysical technology employed in cell counting, sorting, biomarker detection, and protein engineering. It works by suspending cells in a stream of fluid and passing them by an electronic detection apparatus. It allows for simultaneous multiparametric analysis of the physical and chemical characteristics of up to thousands of particles per second. It is often used in studies of cellular diseases and disorders. Fluorescence-activated cell sorting, or FACS, is a special kind of flow cytometry that provides a method for sorting a mixture of biological cells into two or more containers, one cell at a time, based upon the specific light scattering and fluorescent characteristics of each cell. This is useful because it provides fast, objective, and quantitative recordings of fluorescent signals from individual cells as well as a physical separation of cells. Immunohistochemistry staining refers to the process of detecting antigens or proteins in cells of a tissue section by exploiting the principle of antibodies binding specifically to antigens in biological tissues. Preparation of the sample requires proper tissue collection, fixation, and sectioning. Sections can be sliced on a variety of instruments, most commonly a microtome or a cryostat. Antibodies are classified as primary or secondary reagents. Primary antibodies are raised against the antigen of interest and are typically unlabeled. Secondary antibodies are raised against primary antibodies. Using two antibodies is more sensitive and provides more amplification. This is my bibliography. Thank you for your time.